Hello, I'm Will. Welcome to ResearchPod. In recent years, vaping has emerged as an alternative to traditional smoking, with e-cigarette devices forming a key part of plans to reduce smoking rates and associated health risks. Linsey, Haswell and Nathan Gale are part of a team that set out to measure the nature and extent of toxicant exposure and potential biological effects from e-cigarette use. The team compared clinical measurements from exclusive e-cigarette consumers with smokers, assessing the impacts of vaping and consider the harm reduction potential of e-cigarettes in a shifting consumer landscape. For more information on the disclaimers and this study, please visit bat.com and read their original research in the Journal of Internal and Emergency Medicine. And joining me to talk about their work from BAT, Linsey and Nathan. Good morning and hello.
Good morning.
Could you tell us a bit about your work at BAT and how that relates to the research that we're talking about today?
I'm Nathan Gale. I'm Clinical Studies Manager for BAT. I have a bachelor's degree in molecular science and joined BAT almost 20 years ago as an analytical chemist. I've been working in human research for over a decade, and the majority of that in clinical research, designing, leading, and interpreting a range of clinical studies, primarily on our alternative nicotine and tobacco products. And for this particular study that we're discussing today, I was BAT's lead from conception and throughout the clinical execution.
Well, thank you very much for that. And Linsey, could you tell us a bit about yourself as well?
Hi, I'm Linsey Haswell. I'm a clinical scientist here at BAT. I have a degree in genetics and I have worked in academia for 5 years. And very similar to Nathan, I joined BAT over 18 years ago. I originally joined BAT in the in vitro capacity and I worked in a lab for 15 years. And then for the last 3 years, I have been working alongside Nathan in the clinical department doing human-based studies. And I supported Nathan throughout the practical part of this study. I have also been heavily involved in the writing and preparation of the manuscript that we're going to talk about today.
First, I think we should cover some of the background, some of the scope of where this fits into public health, where this fits into personal health, and how timely it is that we're talking about it today.
Of course, if we look backwards, so back in the 1950s, evidence started emerging around the health risks associated with smoking. And BAT's R&D science function was established in the mid-50s, 1956, with an aim to understand these risks and to support the development of less harmful alternatives to cigarette smoking. We now know that it's the burning of tobacco or combustion at high temperature that creates more than 7,000 chemicals, many of which are harmful and have been associated with smoking-related disease. Now, today, surprisingly to many, 1.3 billion people around the world smoke, and that's up from 1.1 billion in 2019. And this is despite the serious health risks that we're all aware of associated with smoking.
That is surprising, considering, well, everything. What role then does your research team have in responding to those numbers?
Clearly, the best way to avoid these health risks would be to not start smoking in the first place or quit. Unfortunately, not all smokers are able or willing to quit. So that's why BAT has created a portfolio of alternative tobacco and nicotine products where people can still consume nicotine but without the burning of tobacco. At our global R&D headquarters in Southampton, we have conducted over 150 studies on these alternative products. And the paper we want to discuss today, that's our first clinical exposure study on our Vuse electronic cigarettes, also known as vapor products.
That leads neatly on to the next question and kind of advancing where this fits into harm reduction. How is harm being reduced and how does that compare to other harm reduction initiatives from outside of tobacco research?
So harm reduction is a social policy and it's aimed at minimizing harm caused from behavior that can be hazardous, but it can't be completely avoided or prevented. So if I take some everyday examples of that, we can't avoid people driving cars or other vehicles or riding motorbikes. But for harm reduction, seatbelts and crash helmets were introduced. And a further example is we'll never be able to stop people unwillingly or willingly being exposed to the sun's harmful rays, but sunscreen was developed to minimize the effects of those things. So the idea of applying harm reduction to cigarette smoking really gained prominence in the 1970s.
So, today, several prestigious health authorities, such as the US Institute of Medicine and the UK Royal College of Physicians, have explored the idea that tobacco, as long as it is consumed not through combusted means, that tobacco and nicotine could still be consumed pleasurably by relatively less risky methods. This comes from a concept back in the 70s suggested by a researcher called Michael Russell, where he commented that people smoke for nicotine but die from the tar. And this is suggesting a potential pathway that eliminates much of the harm arising from smoking combustible tobacco products.
In terms of less risky means, a lot of what we're talking about today is based off of e-cigarettes, which I understand is separate from vaping, and there are other nicotine replacement products. What makes something an e-cigarette versus a vape device, and does that fit under any umbrella of nicotine replacement? What's kind of the categorization that we're talking through here?
So, nicotine replacement products, which are typically patches and gums, but you also get inhalers as well, they first became available as smoking cessation aids right back in the 1970s. And while these NRTs do work for some people who want to quit smoking, unfortunately, they don't work for everybody. This is probably because they don't really provide exactly the same sensorial or consumer experience that people are used to when they smoke every day.
Yeah, and it's important to note that NRT are medically licensed smoking cessation treatments, whereas our alternative tobacco and nicotine products are actually consumer products backed by science, but they're not medically licensed for cessation. We were the first company to have a medical license for an electronic cigarette product called the Evoke, but at the time the license was granted, the product wasn't commercially viable as technology and consumer preference had changed.
So the market demand has changed around that considerably then.
It has, yes. Consumers are generally more accepting of these consumer type products. And as Linsey mentioned, these licensed nicotine replacement therapies, whilst they may work for some, don't actually work for all smokers. So it's important to offer a range of these, potentially less harmful products to smokers.
So we've got a number of different alternative products on the market at the moment. We have modern oral pouches or nicotine pouches, and these are disposable nicotine pouches that consumers place between their gum and their upper lip, and they typically use them for about 20 minutes at a time. And the nicotine and flavors are then released and absorbed in their mouth. We also have tobacco heating products, and these are where we have tobacco containing rods that are inserted into a heating device that generates a nicotine consuming aerosol that people inhale, but it's heated, definitely not combusted. And we also have, vapor or electronic nicotine products. And these are battery powered devices where you have an e liquid that typically contains nicotine and flavoring that then is heated to create, again, an aerosol that contains nicotine and that people inhale that.
With the changing products made available from BAT, going from just burnt tobacco to what is currently available in any future products that do come up, any listeners might have questions regarding what kind of place you have as a tobacco company and as a provider in that harm reduction and the research space around all of that. So, is there any work that you think listeners should be aware of to kind of ground how the work that we are talking about today does have its clinical value, does have its medical utility, does have its important place in literature, and is not just going to be a corporate press release?
So, obviously, the best way to avoid the risks associated with cigarette smoking is to not start or quit. However, not everyone is able or willing to quit. So, that's why here at BAT we've developed a portfolio of alternative tobacco and nicotine products. And these products aim to satisfy and provide a great experience for people without the risk associated with combustible cigarettes. And currently, to date, we have 23 million consumers of these alternative products.
Absolutely. I mean, when you think about a continuum of risk for nicotine and tobacco products, like we've talked quite a lot about combustible cigarettes here, and they're like they're at the extreme end of the continuums of both exposure to toxicants and the risk associated with consuming tobacco and nicotine. And primarily, the reason for that is the generation of toxicants when you combust tobacco. So, what we are focused on is developing a range of different nicotine and tobacco products where smokers who are unwilling or unable to stop using nicotine can potentially switch to these products and use them instead of cigarettes, thereby reducing their exposure to toxicants and reducing their potential risk of developing smoking-related diseases in the future. And we have a robust risk assessment framework which we employ to evaluate these newer generation products. And that runs right through from assessing the chemistry of those products, looking at the constituents in the aerosol, through to preclinical testing and looking at in vitro models and responses to exposure to these new products, all the way through to what we're discussing today, which is clinical data where we're looking at assessment of exposure and potential risk in actual humans. So the gold standard would obviously be long-term epidemiological data. When people switch to using these products, do we see less morbidity, less mortality as compared to when people continue smoking cigarettes? But in the absence of that long-term epidemiological data, there isn't a single study that can be conducted to definitively answer that question about the potential health impact of these alternative nicotine and tobacco products. So we have to take a weight of evidence approach to this. And back in 2001, the US Institute of Medicine recognized the need for this framework to evaluate whether these new alternative products could reduce health risks while offering smokers a satisfactory alternative to continued smoking. And the US Institute of Medicine's report explored key questions to help determine that comparative risk. So, do the products decrease exposure to harmful substances? Is that decreased exposure associated with a decreased risk to health? And are there surrogate indicators of harm other than diagnosed cases of disease that could be measured to gauge the comparative health risk in the short term? And what are the public health implications of that? So, drawing on this work by the US Institute of Medicine, BAT developed a scientific risk assessment framework which has been published and peer reviewed. And this guides our approach to researching the effects of our new alternative tobacco and nicotine products compared to cigarettes.
Nathan, you mentioned that there were the surrogate indicators and all of the decreased exposure. What kind of research and how much of that research has BAT done to find out what can be made better for that public health intervention reason? Linsey, could you tell us more about that?
So yeah, using the framework that Nathan mentioned, we undertake studies on our products that cover emissions, exposure, and risk. And to date, we've published more than 150 studies on our new alternative products. And this body of evidence shows that for our vapor and modern oral products or nicotine pouches, these products emit 99% less toxicants compared to cigarette smoke. And for our heated tobacco products, where they emit 90 to 95% less toxicants compared to combustible cigarettes, we've shown that they have little or no effect on the biology cell types that we've looked at so far. And for our clinical studies for modern oral or nicotine pouches and our tobacco heating products, we have shown significantly better biomarker results for biomarkers that are linked to smoking-related diseases, and when we compare those to the use of combustible cigarettes.
We talk about biomarkers, we talk about surrogate indicators. Again, I think it's important to state some terms and make sure we're starting off with some familiarity of what we're looking at here, and we're going to get into some quite molecular, if not atomic, definitions. So, when we say biomarker in this case, what exactly do we mean, where could we find them, and what do they indicate further down the line?
So when we talk about a biomarker, this is an objective measure that you could look at in somebody's breath, their blood, or their urine. And it captures or tells you something about what is happening to those cells or that organism at that moment in time. So for us in this study, we've looked at biomarkers of exposure and biomarkers of potential harm. A biomarker exposure, a great example of that is an alcohol breath test. So when you consume alcohol, you're then able to detect that exposure to alcohol in your exhaled breath. For a biomarker of potential harm, that is an early indicator of the development of a disease. So an everyday example of that might be increased sugar levels in your urine, and that would be associated potentially with indicating the risk of development of diabetes. Biomarkers can also be affected by a number of different things. They can also be affected by the environment that you live in, the diet and lifestyle that you have, and even the genetics.
Yeah, and in this study, as Linsey mentioned, we looked at a range of biomarkers of exposure, and they were selected from the World Health Organization's study group on tobacco product regulation, who created an initial list of priority toxicants that they recommend reduced in cigarette smoke emissions. So, to give an example of one of the biomarkers of exposure that we assessed in this study, cigarette smoke contains benzene. We can assess the amount of exposure to benzene by measuring its metabolite, which is known as SPMA, in a smoker's urine or in a participant's urine. We also looked at a range of biomarkers of potential harm, and these are early indicators of processes linked to smoking related diseases. And an example of one of the indicators we looked at is called SICAM1, which is a biomarker which we can measure in the blood. And increased levels of that biomarker are associated with a process called endothelial dysfunction, which is a step along the path to the development of cardiovascular diseases. For a couple of the markers we looked at, they kind of fall into both buckets. So NNAL is a biomarker of exposure to a tobacco-specific carcinogen known as NNK. But NNAL itself is also a carcinogen and is associated with the development of lung cancer. So it's also considered a biomarker of potential harm for lung cancer. COHB, or carboxyhemoglobin, is an exposure marker for carbon monoxide. But again, that marker is also associated with decreased oxygen carrying capacity in the blood and, again, the development of cardiovascular disease.
How many people have been examined in this particular study? Some of the sample size and the scope, how far-reaching, how broad and deep are we talking about in investigation here?
So in this study, we wanted to quantifiably assess the differences between current electronic cigarette users and current smokers. And here we had 7 primary endpoints, and they were for biomarkers of potential harm that are associated with the potential development of smoking-related diseases. We also had 8 biomarkers of exposure here, and as Nathan mentioned, they were based on the WHO top Reg 9 toxicant list. And the types of people that we wanted to look at in that study, we wanted to look at healthy, normal adult subjects. So these were people aged between 19 and 55. And then we had 4 different separate populations or groups of people in this study. We obviously had people that use electronic cigarettes, and these were people who exclusively used electronic cigarettes. And they self-reported using the Vuse products ePen 3 and ePod for at least 6 months prior to beginning the study. And we wanted to look at people that were spontaneously using exclusively Vuse products. So these are products that they have chosen and were using in their everyday lives. We also wanted to compare this to obviously current cigarette smokers. So again, these were people who were self-reporting use of combustible cigarettes for at least 6 months before the study started, and they used typically more than 10 cigarettes per day. We also had some former smokers in there. So these were people, again, who'd self-reported quitting smoking for at least 6 months prior to starting the study. And we also had never smokers in there. So never smokers are people who you would also consider non-smoking and had smoked less than 100 cigarettes in their lifetime and had smoked none in 6 months prior to starting of the study.
You mentioned the current smokers there. Is that people who are exclusively using burnt tobacco, or is there some overlap between smoking cigarettes and also smoking any electronic device?
Again, these were people who exclusively used combustible cigarettes and didn't use any other nicotine or tobacco containing products.
Okay.
Yeah, as Linsey mentioned, in our groups of exclusive Vuse users and exclusive former smokers, we wanted people who had been exclusively using Vuse for at least 6 months. And in the former smoker group, people that had quit smoking for a minimum of 6 months. Now, when we brought these people to the clinic, we looked at cotinine in their urine, which gives us an indication as to whether they're actually a nicotine user or not. And we also looked at exhaled carbon monoxide, which gives us a further indication as to whether they are actually smoking combustible products or not. And it was really important that this was verified, because if we had people who had told us they'd quit smoking or were exclusively using Vuse, but they were actually smoking cigarettes, this would have quite a large effect on the study data. Now, the urinary cotinine and exhaled carbon monoxide measures that I mentioned are quite useful, but they're quite short-term measures, and they certainly wouldn't cover the previous 6 months. So what we used in this study was something called cyanoethylvaline, or CVAL for short, which is a blood adduct of a cigarette smoke toxicant. So we can measure this in the blood. And because it's an adduct which sticks to red blood cells, it takes around 120 days for all your red blood cells in your body to be cleared and replaced. So that marker in the blood gives us a really good indication as to whether people had been smoking for a longer period of time, and just adds to that confidence that the people in the views group and the former smoker group genuinely not been smoking for at least 6 months. So we recruited 213 participants for this study, which took place at a clinic in central London. And when we got to the end of the study, we had 205 participants who completed the study and were compliant with that blood CVAL measure that I just mentioned. So we had a good split of female and male participants in the study, around 41% female and 59% male. And the average age of the subjects was around 31 years old. In the e-cigarette and views user group, we had 93 participants complete. We had 40 smokers complete, 35 former smokers, and 37 never smokers.
Okay, well, you've mentioned the population and the location in terms of the schedule and the rigmarole of the assessment and all of the samples that were collected. Was there any particular methodology, any procedures that were being followed? And from there, we can lead into the results of what that all found.
Yes, so the design of the study and the procedures that participants went through, I can give you a brief overview of that. So on day one, so the first day of the study, potential participants are invited to attend the clinic in central London to undergo what we call screening. This involves a variety of physical tests and assessment of their medical vital signs, looking at alcohol, drug, and pregnancy testing, and also confirming nicotine use and smoking status using those measures I mentioned before, exhaled carbon monoxide and cotinine, and also applying questionnaires to look at people's extent of tobacco and nicotine use as well. And this screening is designed to achieve two purposes. Essentially, first of all, to make sure it is safe for that potential participant to actually take part in the study. So the findings from that screening assessment are reviewed by a medically qualified person at the clinic. And secondly, to make sure that the subject is appropriate for the scientific means for the study. So after that screening had been performed on the same day, individuals who met those inclusion criteria were enrolled immediately and began to participate in the study. So they were kept in the clinic. They supplied their own electronic cigarettes or combustible cigarettes, depending on which group of the study they're in. They were asked to bring sufficient numbers of those to cover their typical use for an entire 24-hour period. And they were allowed to use them as they typically would use those products. Throughout their 24-hour period there in the clinic, we collected all of the urine that was voided by each of the subjects, and we performed blood sampling. And these were the key biosamples for looking at biomarkers of exposure, potential harm, and also the hemoglobin CVAL adduct. We also performed some physiological assessments and had our participants complete a quality of life questionnaire as well. So then at the end of day two, there were some final health and safety assessments to make sure nothing significant had changed with the participant during the study. And then the participant was discharged from the study. And then a follow-up phone call was performed with that participant around 7 days later, just to make sure they hadn't had any adverse experiences since they'd been discharged from the site. Those blood and urine samples that we collected were then transported to our partner labs. So the majority of the biomarkers of exposure in the study were analysed by a lab called Analytische Biologisches Forschungslabor, or ABF, who are based in Planegg in Germany. And the biomarkers of potential harm, some were performed by ABF again, others performed by Solarion, based in Zurich in Switzerland, and a couple of the markers were also assessed by a local medical laboratory called the Doctors Laboratory, based in London.
And with all of that in mind, how does smoking, electronic cigarette use, prior exposure, never smoking, how do they all compare? Is there anything that is markedly different, better, worse, remarkable? What's the results here?
So the results of our study have shown that views consumers have favourable changes in biomarkers of exposure and biomarkers of potential harm when you compare those compared to those who smoke. Particularly for the priority to smoke toxicants that we looked at, or the biomarkers of exposure in this study, all of the biomarkers of exposure, that was 8 that we looked at in the study, had lower levels and these were significantly lower as measured by stats when you compared the Views users to smokers. For the biomarkers of potential harm, we saw favourable changes between Views users and the smokers across all of the 7 that we measured in this study. We actually saw statistically significant changes in 3 out of those biomarkers of potential harm, and they were actually related to cardiovascular disease. So we saw favourable changes in 11-DTX, which is a biomarker of platelet activation. We also saw statistically significant changes in carboxyhemoglobin, and this is a biomarker that is related to the oxygen carrying capacity of red blood cells, and again related to cardiovascular disease. And we also saw significant reductions in soluble ICAM, which is an endothelial dysfunction biomarker, again linked to cardiovascular disease. We also saw a significant reduction in NNAL, which is a biomarker of exposure, but also a biomarker of potential harm because it has been linked to the development of lung cancer. And as Mason mentioned before, we also did some physiological measures and some surveys as well on our participants. In particular, we had done a quality of life survey, and we actually saw some favourable changes in the general health outcome there, or item on that questionnaire, which also was very encouraging for us. Also worth noting as well, because we looked at 7 markers here for biomarkers of potential harm, we also did some statistical adjustment for multiple comparisons, which, because we looked at 7, we also wanted to decrease the chance of a false positive here in this case as well. So, we did some robust statistics in this study as well.
Yes, you mentioned carboxyhemoglobin, SICAM, NNAL. Those are all ones that we highlighted earlier. So out of the significance of those thresholds, how significant are we talking? Again, you mentioned the difference between absolute risk and measured risk.
So if we take NNAL as an example, as Linsey mentioned, there were significantly lower levels of NNAL measured in the Views users compared to the levels of NNAL measured in the smokers. And actually, those levels of NNAL in the Views users, the numbers were not that different to what we saw in the former smoker and never smoker group. So that really indicates that these Views users' levels aren't just slightly reduced compared to smoking. We're really getting down towards the levels of people who have completely quit smoking for at least 6 months, and even potentially towards those levels that you would see in people who have never smoked.
And with the trial design, you mentioned some of the structure and the limits of it as a kind of comparison to any other investigations there might be that looking maybe long-term or cross-sectional. Is there anything that you think is going to be an advantage of how this study's been conducted, or any questions that it raises for future investigation?
Yeah, so big advantage of this study compared to sort of longitudinal studies is this study involved a single clinic visit. It really captures the lived experience of people who are using these products, with, as Linsey mentioned earlier, the participants self-selecting use of these products and using them in the way they want to in their everyday lives. That's quite a stark contrast to a longitudinal switching study where you would bring in someone who is a smoker and then you would force them to switch to using your specific product and look at the change in that individual over time. So in this study with the single clinic visit, what we were assessing is the difference between groups of participants. So we're not controlling for baseline when these people were perhaps smokers, compared to a longitudinal study where you're actually looking at differences within participants and changes from baseline. So a limitation or a challenge with this sort of single cross-sectional type study is it increases the potential for large variability because we're not controlling for baseline. So the findings we've actually achieved in this study, in my opinion, are all the more impressive given that risk of greater variability. Big advantage of running a study of this type, as I mentioned, single clinic visit. The study can be run a lot quicker. It took us around 1 to 1.5 years to complete this study end to end. Whereas with a switching study, depending on the switch duration, you're looking at around 2 to 3 years to complete this type of study. And these studies, that's really important. Duration of these studies is really important for consumer products in this really fast evolving environment where new technologies are being developed on a very, very regular basis, and consumer preferences are in a state of change as well.
So in this study, we wanted to assess the differences between current exclusive electronic cigarette users and current smokers. And we looked at biomarkers of exposure and biomarkers of potential harm. And what are the results of these studies have shown is that consumers of BAT's Vuse, ePod, and ePen 3 electronic cigarette products have shown better results for the biomarkers of exposure, the 8 that we looked at here, which are linked to this smoke toxicant generated by the WHO. And we also looked at biomarkers of potential harm, and we saw significant changes in 3 out of the 7 that we looked at when we compared users of electronic cigarettes compared to smokers.
Yes, this is a really positive set of results, and we strongly believe that the results we've got here underscore the contribution that vapor products can make towards tobacco harm reduction. And so the study adds to an already significant and constantly growing body of evidence around vaping potentially being a less risky alternative for smokers who would otherwise continue to smoke.
And if there's anyone listening to this from a consumer background, from a healthcare provider background, from a research background, is there anything that you think they should come away from this interview knowing, or any next steps that you think would be useful for them to take?
We really think that these results show that electronic cigarettes have a role in tobacco harm reduction. And we think this study shows that our consumers should have high confidence in BAT's well stewarded products and that they're backed by science. We do the research and we publish this. We also think it'd be really good if regulators and policymakers also considered our results in achieving their objective of reducing smoking prevalence in countries. I also think these results are important for the wider scientific community as well, and other people in related industries, such as, other important players in the tobacco industry.
And if people want to know more, either to find out more about the detail of this study or find out more about any future research from BAT, where can they find that and where can they find you?
So our paper has been published in the Journal of Internal and Emergency Medicine. It was published on the 30th of May, which is actually World Vape Day. And if you go onto the journal website, we are actually in a special collection, which is being compiled. There's a number of different papers there. The title of that one is The Health Impact of Electronic Cigarettes and Tobacco Heating Systems. And our publication is open access, so anybody can click on it. It's free to download and read.
Yeah, as an organisation, we're really keen on openness and transparency, and we publish the findings of our research on our science website, which is bat-science.com. And on our corporate website, bat.com, we also have a feature from our Director of Research and Science, Dr. James Murphy, covering this study and his take on what it means for tobacco harm reduction. So I encourage people to visit the corporate website and view James's thoughts.
And if anybody has any questions that they'd like to ask us, you can certainly contact us through Bat Science, but our contact details are also in our publication if you want to download and contact us about any questions or comments you might have. We always look forward to talking to people about it.
Just want to thank you, Will, for hosting this podcast. Myself and Linsey really enjoyed discussing this key study, and we really hope it's of interest to your listeners.
Thanks so much for your time, and speak with you again sometime soon.
Thank you very much.
Absolutely. Thank you.